we're going to talk about how hair is useful to forensic scientists. So what are the parts of hair, and then what are the parts that we can analyze to try to compare hairs found at the crime scene. So the basics first is the hair shaft. The hair shaft is composed of 3 parts. Okay, the cuticle, which is the outside covering made of overlapping scales. The medulla, which is the inside layer running down the center of the cortex. And then the cortex itself, which is kind of like the inside. So it's not the very outside and it's not the middle. It's kind of the filler part. It's made of keratin and it's got your pigment. Those are the 3 parts of hair. Okay, so your cuticle, your medulla, and your cortex. So let's take a look at each one a little bit more in depth. So the cuticle is first. Your cuticle, like we said, is the outermost layer of your hair, so it's the very outside edge. It's covered with scales. The scales always point towards the tip of the hair, and there's 3 basic patterns, coronal, spinous, and imbricate, and you're going to want to jot those down in your notes. Try to make them a little bit bigger for you so you can see. So this is one of the characteristics that you can use to compare hairs, is you can figure out what type of cuticle scale they have. Coronal scales look kind of like cups sitting on top of each other. They're oftentimes found in animals. The second type is spinous. Spinous cuticle patterns, they kind of look almost like an artichoke, like a whole artichoke where you kind of have layers that you peel back. So this is what a spinous cuticle pattern looks like. And then the third pattern is imbricate. And imbricate almost looks like if you used a paper mache, and you have to put like all the little pieces of paper kind of over top of each other. Kind of looks like a bunch of little cracks on top of each other. That's what imbricate is. And animals can have all 3, coronal, spinous, and imbricate, but humans only have imbricate patterns. There are some animals that also have an imbricate scale pattern, but humans only have imbricate scale patterns. So if you find a hair at a crime scene and it's got an imbricate pattern, you can investigate it further to see if it's human. If you find one that is spinous or coronal, you know that it's for sure some type of animal hair. The second part of hair is the cortex that we're going to talk about. And the cortex is the part of hair that gives hair its shape. So whether it's like curly hair or straight hair or kinky hair, it gives its hair its shape. The other thing that's found in the cortex is your melanin. The melanin is the pigment granules that give your hair color. Okay, so if you have brown hair, you have brown pigments in your melanin. So the cortex is right here. It's the inside part, not the very middle, but kind of that inside filler part. Gray hair is hair that has the absence of any pigments. So if your hair turns gray, it means there's no more melanin or no more pigments that are there. So if you have blonde hair, if you have red hair, you're going to have different kinds of pigments, different colored pigments. Gray hair is the absence of any pigmentation. And then you also have the cortical fusi, which are air spaces. They kind of look like little pockets, and you can see they're right here. Okay. Most of the time they're found closer to the roots of the hair, but sometimes they can be in the middle. So the main thing to remember with the cortex is it gives your hair its shape and that it has the melanin or the color for your hair. The third part of hair is called the medulla. The medulla is the inside, the very, very, very inside of your hair. It's the core of the hair. It's not always visible, and it comes in different types and patterns. So when we talk about the medulla, it's the very inside. So right here is a medulla. Okay, and there are 5 patterns. Fragmented, which looks like this. So if this is your hair, it's where the medulla is there sometimes, and then there's long stretches where it's not there, then it's there, then there's long stretches where it's not. Interrupted, which is where it's there more often than not, but there's some spaces where it kind of breaks up. Continuous is where you have one long medulla. Okay. Stacked, okay, it's like, it kind of looks like little circles all stacked on top of each other. And then the 5th pattern is absent or not present, and that's just where like you have no medulla at all. Human hair, the medulla can be fragmented, it can be continuous, or it can be absent. Those are your three options. Human hair won't be stacked, and it usually won't be interrupted as well. It'll usually be fragmented, continuous, or absent. Another useful characteristic, besides just figuring out what medulla pattern the hair has, is to do what's called to calculate what's called the medullary index. The medullary index is taking a look at the width of the medulla compared to the width of the entire hair. And a rule of thumb for this is in human hair, the medullary index is generally less than 1/3 of the hair. And in animals, the medullary index is usually greater than 1/2, which means that humans usually have a skinnier medulla than animals do. Animals usually have thicker medullas. So how this works is you take your ruler, you line it up, and you figure out, okay, how wide is my medulla? So we'll say that it is 0.5, okay, and then 0.5 centimeters, and then compared to the width of my entire hair. And so let's see, let's put that in a different color. So we'll take a look here and we'll say that the whole hair is probably 1.5. So you do 0.5 divided by 1.5. So 0.5 centimeters divided by 1.5 centimeters, and you'll see when you divide that, your medullary index is 0.33, or in this case, it's 1/3 if you turn that into a fraction, which would indicate that this is most likely human hair. Animal hair, it's usually greater than 1/2, so it'd be 0.5 and above. Okay, and a lot of times when you're looking under the microscope for the medullary index, you have to just estimate. Like this one, is it 1/4 of the whole hair? Is it 1/2? Is it 1/3? So on and so forth, because sometimes you don't have a ruler that can get underneath the microscope. So the medullary index is one way that we can use to compare hairs. We can compare what the medullary index is. Alright, so let's take a look at some useful characteristics. So we've already talked about cuticle pattern, we've talked about medulla type, and we've talked about the medullary index. Now we're going to take a look at some other, besides just the three parts of hair, what are some other characteristics that are useful when analyzing hairs that are found at crime scenes. One of the things we can take a look at is hair growth. What stage of growth the hair's in. So there's 3 stages of hair growth. The first stage is called the anagen stage. The second stage is called the catagen stage. And the third stage is called the telogen stage. So let's look at stage 1 first. That is where the hair is actively growing. So if your hair is actively growing, it's in the anagen stage. Hairs will stay in this stage for up to 5 years. And if a hair is pulled out during the anagen stage, it will have what we call a follicular tag. This is important. A follicular tag are skin cells that are attached to the root of the hair that still can contain DNA. So right here is a follicular tag. So you can see this is the hair right here. Okay, this is the root of the hair down here, and you can see that there's almost like a complete layer over top of it. Okay, that is the follicular tag. Those are skin cells that got pulled out when that hair got pulled out. Here's another example. You can see here's the actual hair, and then all the way around it, okay, those are skin cells that got pulled out. Those are a follicular tag. So if it's in the anagen stage, it's actively growing, and it gets pulled out, it will have the follicular tag attached to it. If it's in stage 2, it's the catagen stage. That's where the hair is decreasing in growth. This is what we call a resting stage. It's not really falling out yet. It's not really growing anymore. The root appears to elongate, so it starts to elongate down here. It looks, instead of being like bulb shape, it starts to kind of stretch out a little bit. And then the third stage is the telogen stage, where the follicle is getting ready to push the hair out of your head. This lasts anywhere from 2 to 6 months, and the root starts to look like a club appearance, so it's pretty flattened out compared to a bulb-like appearance. So knowing which stage the hair in is nice, because if it's in the anagen stage, then you're looking for a follicular tag. If it's in one of these 2 stages, it's possible that the hair was transferred and not ripped out. Another thing that's nice to know is that hair grows about a centimeter a month. Okay, so if you find hair at your crime scene and say it's 3 centimeters from the root, there's color in it, well, it means it's been about 3 months and the person got their hair colored. Different information like that, it's always valuable to know, especially because your hair soaks up all sorts of toxins you put in your body, so like drugs and things like that all get sucked in your hair. So they can kind of give you a timeline of drug use from your hair based on growth and where it's absorbed what. The root is also a useful characteristic. Again, we kind of talked about whether it had fallen out or was forcefully removed. So this is a hair that had fallen out, and then this is a hair that was forcefully removed. And you can see there's a follicular tag on this one, and you can see it's very jagged, and that's what shows that it was forcefully removed. If the hair has been forcefully removed and some of that follicular tissue is on there, you can try to do a DNA match or sample from there as well. Hair itself, by itself, doesn't have any DNA, but if you get the follicular tag on it, you can test for skin cells with DNA. So how do we get the hair? To collect hair from the following individuals, you always want to collect from the victim, any possible suspects, and anybody else who may have deposited hair at the scene. So if there were like dogs or animals or anything like that, you would want to collect their hair as well. Here's a proper sample. Okay, a good control sample includes 50 full-length hairs from all areas of the scalp. So they'll pull 50 full-length hairs from kind of all over, the bottom, the top, and then if needed, they'll do 24 full-length pubic hairs as well. So if it was a sexual assault case or anything like that, if there's no signs of sexual assault, oftentimes they won't do the 24 full-length pubic hairs. But if there were any signs of that, they would do that as well. So to recap, so now that we have the hair, we've collected it, here are all the comparisons. So if you had an unknown hair, say from the crime scene, and you had hairs from your suspects, here is what they are going to take a look at. They're going to take a look at color and length, the distribution, shape, and color intensity of the pigment granules, like brown, red, blonde, so on and so forth, the scale types of the cuticle, whether or not there's a medulla, and if there's a medulla, what is the medulla pattern and what is the medullary index. And then if it's dyed, if the hair has been dyed in the cuticle and the cortex, what does it look like? And then the root, to take a look at the age of the hair and the follicular tag. So those are some of the things that they are going to take a look at and compare. Hair is only class evidence. Okay, hair is not individual. You can help place a suspect at a crime scene, you can exclude perpetrators, and you can sometimes indicate race depending on the shape of the hair, but that's about all you can do. So you can say, okay, well, I had 3 suspects, I had a blonde, I had a brown, I had a red hair, and at my crime scene I found blonde hair. So you could automatically exclude your brown hair and your redhead, and you could say most likely this person was there, all their hairs kind of matched, but you can't say for certain that they committed it because there's all sorts of other people that could have those same characteristics. Unless you find a follicular tag on a hair and you can run a DNA match, hair is still considered class evidence. So it can't prove a suspect committed a crime because it's not an individual piece of evidence, it's only class, but it can significantly narrow down your search and it can be very probative in court. The last thing that you can do is you can run toxicology on hair. It's nice because hair is easy to collect and store and it's externally available. They take hair from all over the body to get an accurate timeline, and they can get a pretty good idea of an individual's history of drug use or evidence of poisoning because your hair sucks up everything that goes into your body and it stays in your hair as it grows. So it can say, this person stopped using drugs approximately 6 months ago, or this person is using drugs up until current time, so on and so forth. They can do pretty good toxicology screens via hair.